I'm pleased to give a warm welcome and introduction to Brett Howard, biologist with the Department of Fisheries and Oceans. Brett Howard began her academic career at the University of Alberta before going to Dalhousie University in Halifax, and rounded off her Canadian tour of universities by completing her PhD at Simon Fraser University. Her thesis work focused on the spread and impacts of invasive marine crabs, focusing on European green crab. Currently, Brett is continuing her work on invasive species as a biologist at the Department of Fisheries and Oceans. And at this point, I will pass the mic over to you, Brett, and I hope you all enjoy the webinar.
Hi, everyone. Thank you so much for attending. I'm going to share my screen now. And yeah, that's coming through, right? Yes, good. All right. Hi. So thank you so much for having me today. I'm very excited to talk to you about invasive marine crabs in general, as well as my particular organism of interest, which is the European green crab. I'm going to just jump right in. I'm sure as people who are familiar with and interested in invasive species, you've heard of the 100 worst invasive species list as put out by the International Union for the Conservation of Nature. Looking at all of these little pictures, you might notice some trends, lots of terrestrial species, plants in particular, plenty of invertebrates as well, but mostly insects. However, there are quite a few invasive aquatic species and a few marine ones that we are particularly concerned about. Only 2 of the species on this list are in fact crabs, the European green crab, as well as the Chinese mitten crab. The Chinese mitten crab is partially freshwater, partially marine, and I'm going to talk about it just a little bit in this presentation. But what I want to start with is just to say that although this list is very useful for many reasons, it is by no means comprehensive. So marine crab introductions are on the rise globally and have been for a very long time. Part of this is because, of course, we're just recording more species and we're paying more attention to new species in areas where they don't belong. But this is also because people are moving around more, and where people go, so do species. So this is the cumulative number of recorded, which means actually detected, which, you know, there's lots that go undetected, of marine crab species introductions worldwide up until about 2015. I'm going to structure this talk around the invasion process. The invasion process is a nice way to think about how we get from the movement of non-native species all the way up to the impacts of non-native species. And this is just, for me, it's a nice way to structure the talk. So I'll be going through all of these in some in more detail than others. I'm going to go through transport and establishment relatively quickly, talking about marine invasive species more generally, and then I'm going to get into spread and impacts, which is where our research is more focused here at DFO. So the transport of invasive species in the marine realm is largely inadvertent. There are a few vectors that we are particularly concerned with, ships being the biggest one. If you think about the stuff that grows on the bottom of hulls, especially on a ship traveling internationally, the number of species that can be on the bottom of that hull that are potential invasive species is tremendous. Even more challenging is ballast water. In ballast water, the species are usually in a planktonic or larval form, and therefore even harder to screen for or much less pick out or get rid of. We also have unintentional transport in aquaculture. This is probably the means by which invasive European green crab got to the west coast of Vancouver Island, or pardon me, the west coast of North America in the 80s. So green crab were already invasive on the east coast of North America, and then probably in packing material or with a shipment of live oysters, there were just little baby green crab mixed in there. And when the oysters were unpacked on the west coast, all of that detritus was just chucked into, the San Francisco Bay estuary, and green crab took off from there. So, I mean, inadvertent, but certainly with significant consequences. And then very occasionally you get the intentional but not always legal transport of marine invasive species. So every couple of years, somebody attempts to smuggle Chinese mitten crab into British Columbia for food and commercial purposes. And this is illegal to do, and it results in fines. And there are always interesting stories because people use very creative methods to smuggle these crab species in stocks and whatnot. And the reason that this all, the common denominator for the transport of marine invasive species is shipping and people. People are, increasingly living in coastal areas. We increasingly have more and more international shipping. This figure shows shipping lanes, I guess, around the world with thickness indicating the volume of ship traffic. And you can see there are areas where there's a great deal of ship traffic, and in some places really congregating in space. So the Mediterranean is a big one, and of course the Eastern Seaboard is another. And if you take this and then map onto it any introduced crab species, so records of crabs being introduced, you can see that it does sort of match up where you've got a lot of ships and a lot of people, like Europe, Mediterranean, you have way more introduced crab species. So this just demonstrates the role, the linkage between where people and vessels are moving and the rate at which we are seeing introduced marine species, including crabs. However, introduced does not mean established. Species can be introduced and then fail to take off in an area for many reasons. This shows, it just says pie charts, the number of those introductions that then turned into established crab populations. And you can see it's actually quite high, right? There's over 50% in most of these locations. And that speaks to the generalist nature of a lot of these crab species. They can live in a lot of environments, they can eat a lot of different types of prey, and they're just generally quite robust. So they have no problem getting established in these non-native environments. Establishment, to define it, requires stability, population stability, and population growth. In order for that to occur, you need to have enough individuals introduced such that you can have a breeding population that is genetically diverse. And they also have to land in a suitable habitat. But like I said, a lot of invasive species, including crabs, tend to be very generalist species. They can live in a lot of different habitats, eat lots of different things, etc. And that's very much the case for European green crab. So this is a European green crab. The picture of the live one is about the average size for female green crab in British Columbia. So that gives you a sense of how it matches up against like our very little shore crabs and then our very big Dungeness crabs. One thing that I really want to emphasize with green crab is that it is very much an intertidal species here in British Columbia. Specifically, we find it in soft sediment, estuary type habitats. Elsewhere in the world, they definitely occur in rockier habitats, but here they tend to be restricted to these more soft sediment habitats and in the intertidal. So when people think crab, they think throwing out like a Dungeness pod at like 15 meters or whatever. We're definitely talking about an intertidal species here. For ID, and I'm happy to come back to this at the end just to reiterate, but for identification purposes, you're looking for the 5 spines on the side of the carapace and the 3 spines in between the eyes. Those are diagnostic. Other characteristics that might help you are the flattened back leg. It's a swimming leg. We have a few species with a semi-flattened back leg, but it's really pronounced in green crab. They also have heteroculus claws, which means they have a smaller cutter claw and a much bigger crusher claw. It's hard to see in these pictures, but that can sometimes help with identifying green crab. What you shouldn't use is the color. Green crab, although called green crab, can range from brown to yellow to orange to red to green. So you shouldn't use color, shouldn't rely on color, not least because we have our own crabs, native crabs that are also green. You don't want to confuse the two. So I'm happy to come back to that, but that's green crab in British Columbia. However, they are established globally. I already kind of explained this a little bit, but they've been on, for example, the east coast of North America for like 200 plus years, and then Australia, followed by South Africa, Japan, and then into Canada. And on this map, I also have all of the failed introductions, just coming back to the idea that introduced does not necessarily mean established. So that's the range of green crab globally. In black, I should say, is their native range, which is Europe and parts of Northern Africa. So that covers off the transport and establishment elements of the talk. And like I said, I wanted to put the bulk of the talk in the spread and impacts part, because that's where the bulk of our research is these days. So for spread, the sort of first question that you want to ask is, how does your organism move? And that's going to obviously will differ quite a lot from species to species. And it also really will dictate how you have to think about their spread, how you have to think about detecting them, and how you have to think about managing them. So an organism like the starling is not really restricted by anything. It can fly. So it's not got huge geographical barriers to contend with. And so over time, you're just going to see this outward ripple effect as they move throughout, the continental United States and into Canada. Whereas a species like the Chinese mitten crab, which as an adult is confined to freshwater habitats, in order to track its movement or predict where it might spread to, you're obviously just going to follow where the rivers are. It can't crawl over land, and so you know that that's where the mitten crab are going to spread. You can kind of predict that, you can forecast that, right? So with green crab, the main way in which they spread is through larval dispersal. As adults, they're more or less restricted to the intertidal habitat in which they live. They're not crossing huge deep water environments. So they're mostly restricted to their little, patch of beach. But as larvae, they can move very far, very quickly. And in our case of British Columbia, it's predominantly a northward spread because it's dictated by currents. The larval duration can be quite long. It's temperature dependent, but 59, 60 days at 12 degrees Celsius. You can get pretty far in 59 days if you catch a good current and you've got enough food and you don't get eaten. And indeed, the habitat suitability for green crab larvae and adults extends all the way up the coast into Alaska. In 2019, plankton sampling in the Prince Rupert area did detect green crab. So we know they're getting at least that far. Finding the source population is much harder and potentially not possible. But we do know that this follows our prediction of just an onward northward spread of green crab. This map shows all of the detections of green crab in British Columbia since DFO started regular tracking in around 2006. So every summer we go out and we set traps and we try to find green crab. They've been on the west coast of Vancouver Island for a very long time, almost 2 decades now. So they got to BC from the US in 97, 98. It was an El Nino year. It was really good conditions. Green crab, lots of green crab larvae were able to seed basically the entirety of the west coast of Vancouver Island. And there they stayed for quite some time. But again, that northward spread that we expected just started to creep up the central coast. And so we have some populations along the central coast. And then we didn't see anything for a little while. And then in the last couple of years, we've seen 2 what we're calling invasion fronts, or what you could call an invasion front. So we have one in the Salish Sea, and we have, as of this year, one on Haida Gwaii. So these are adults, not larvae. I should just clarify. These are we've trapped adults in all of these locations that show up in green. In black, we have places we've trapped but not seen green crab. Now the good news about the Salish Sea and Haida Gwaii invasions is that they are not yet really established. We're not seeing population growth. We're just finding the occasional one on various beaches. And so that means there's still time to do something about this. We can be very proactive in tackling the spread of green crab in these areas. Okay, so when I say we, what do I mean? Before I go any further, I just sort of think it might be useful to outline that. So within DFO, there are kind of 2 branches, if you will, that have their fingers in the green crab pie, so to speak. That's a gross mental image, but there you go. So I'm coming to you from the perspective of DFO science, and our job is to conduct research that informs management, DFO management, which is housed in the fish habitat protection branch of DFO. So we do research that informs what they do. And then we also have lots of clients outside of DFO. And this is a very short list of some of the groups that we work with on green crab issues. And so again, I'm coming at it from this science perspective, but we also have this whole other group of really awesome individuals who are doing the management. That includes, training, that includes education and outreach, that includes lots of the public interfacing, and leading a lot of the rapid response kind of efforts as well. It is a team effort for sure, and I wanted to pull this figure in, not so much to pick through it in detail, but just to demonstrate how science and management work together in an instance like the rapid response that we did in Haida Gwaii. So like I said, this summer, July 2020, they found green crab, a local found green crab in Skidegate Inlet. And immediately after, we convened a technical working group that had already existed. This included the Council of the Haida Nation, Parks Canada, Government of BC, as well as us. And we sprung into action, and very quickly we were able to get traps up north, and they were able to start doing some really intensive surveying to try and figure out where green crab were. I just have to say it was really quite something to see, especially because rapid response is not typically a virtual activity. So 2020 had a whole bunch of challenges, and certainly not being able to be on the ground to do rapid response was difficult. I also want to draw attention to what I've got circled there in yellow. As a science, one of the science roles is to identify the species, confirm the species ID. That was easy. And then we have to do something called confirm the location. And that sounds easy, but it's really, really challenging. And the reason is because detection of any species, especially a rare species like a new invader, is extremely difficult when you're talking about a complex environment. And there are few environments more complex than the near shore of British Columbia. Think about walking along a beach. You go from rocky intertidal to a nice little pebbly cove to a long, flat, sandy beach, in not very much space at all. And some of those habitats are great for green crab and some are terrible. And then added to that, you've got long, long fjords, you've got tons of little islets. And so when you say to somebody, okay, take these traps and put them in a cove somewhere, they're going to go, which cove? And so we spend a lot of time thinking about how to improve our detection so that we can make that eradication trapping or detection trapping more efficient. And the reason that's a concern is because I keep saying trapping, and I should explain what I mean by that. Trapping, in this case, is a very labor-intensive process. It means taking these folding fukui traps out. We like them because we can get quite a few of them on a boat, head out and start hitting beaches, and we have to hit them at a particular tide, bait them, set them, come back 24 hours later, pull them up, and then count everything that's in there. And in some sites, that can mean a lot of counting. Hopefully not, but sometimes it means a lot of counting. And then if that's not working, or if that's missing, say, a certain size class of green crab, we might also want to actually hit the beach at low tide, because like I said, it's an intertidal species, and start picking through rocks and looking under things to see if we can find them that way as well. So this is all very time-intensive work to do this detection trapping. And so it would be very useful if we can make that as efficient as possible for lots of reasons. So this is the question of how do we improve detection? And we just want to introduce the idea of the detection threshold, the idea that at some point, once a species reaches an area, it has to get to a certain number above which we can find there are enough of them that we see them. And the goal here essentially is to move that detection threshold, or lower that detection threshold. We don't want to be only finding green crab when there are hundreds and hundreds of green crab at a beach. We want to detect them when there are very few. So how can we get to that point? And the complicating factor is what happens if you go and you put traps in the water and you don't find any green crab. Does that mean that the larva just didn't get there and there are no green crab there? Does that mean that larva did get there, but the green crab failed to establish and there's something about that habitat that isn't sufficient? Or is it actually a failure of detection? That you have not surpassed the detection threshold, there are green crab there, you just haven't found them yet. So these are all complicating factors in how we understand and predict spread. There are a few really quantitative ways that we go about trying to predict spread and improve our detection of green crab. One is habitat modeling. In particular, we're interested in sites that could become potential hotspots. And when I say hotspots, I'm referring to the really interesting thing about green crab is that they can occur in really like kind of average densities, like Main Bay. In an average set, you get like 200 crabs in a set of traps. Move just a couple of kilometers over into Pipestem Inlet, and a single set, you'll net over 1,000 crabs. So what is it about these two sites that makes them so different for green crab? And so identifying these hotspots we're particularly interested in. And then another thing that we're interested in is trying to figure out how to do all of this quite quickly, because when you have a rapid response situation like we did in Haida Gwaii, you don't want to be spending a ton of time trying to come up with a method for where to go trap when you really need to be on the ground as soon as possible. So we spend quite a bit of effort doing species distribution modeling. I'm sure that you have seen many species distribution models in your time, maybe without even knowing it. And they're excellent. There's several approaches that we're undertaking right now that are either published or in prep as we speak. They're really good for a lot of reasons. First of all, they give you really high coverage, right? You can get the whole coast of BC in one map and look at all of it at once. They're really quantitative, they're really defensible, and they're predictive. So for example, this figure, the species distribution map for green crab is not actually a current, it's not for now, it's for what we predict it might look like in 2075. So taking into consideration climate change, which is going to be good for green crab. Most likely, they are going to thrive in warmer waters, and we're going to see more of them doing well up north, as well as in the Salish Sea, actually. So they're predictive in both space and time. So they're really excellent for all of those reasons. However, they are complex. They take a lot of expertise to design and build, and they take a lot of time to design and build. They also aren't site specific. So if I gave you this map and said, okay, go trap some green crab, you go where? I need way more specificity, and I need it right away. So these are two things that we found. I mean, we knew these were drawbacks of these more complex species distribution models, and it was really highlighted when we went into this rapid response in Haida Gwaii. And so myself and my supervisor, Tom, started working on a rapid site selection method, which is species distribution modeling in a really speedy way. So looking at very few predictors in a very discrete area and trying to just find the exact beaches that we think you should go hit as quickly as possible. And so this is something we're still working on. This is not finished yet, but we've used the sort of rough draft of it a few times, and it's worked really well for sort of directing efforts in discrete areas. But what if you just got rid of trapping altogether? To be fair, we'll never get rid of trapping altogether, but maybe there are ways we can supplement trapping. In a good day, we can maybe trap 2 or 3 sites. So how can we increase that? How can we get to way more sites in a day? And one possibility is eDNA, which stands for environmental DNA. Basically, you're taking a water sample, it's got plankton, it's got feces, it's got bits of detritus in there. All of that DNA is scooped up in one water sample. You barcode all the species, and you figure out what's in that water, what bits are floating around in that water, and that gives you a species ID. And you know that that species is in the area because bits of its DNA are floating around in that water. And the nice thing is that it's going to be really targeted, so you can look for a single species very quickly. It's much less labor intensive in the field. You're just doing a water sample. And that means you can get much better spatial coverage for the amount of time that you're spending in the field. So eDNA has a lot of potential. We're working on making it as robust as possible. So we're doing eDNA water samples paired with trapping to see how the abundance of green crab is reflected in the eDNA sample. And they're actually it's a much better relationship than I would have expected, in fact. So if you get lots of DNA in the water sample, it probably means that you've got more green crab at a site. So this is something we're still working on and refining, but I think it has a lot of potential for speeding up our detection of green crab. So I'm usually not one for summary slides, but that was just a lot of stuff. So really quickly, I just want to just give you a quick summary of everything I just said before we move on to impacts. So green crab spread through plankton, and we expect this northward range expansion and further range expansion in the Salish Sea. Responding to that spread is a collaborative process, and we've had lots of early success in the Salish Sea and in Haida Gwaii under less than ideal conditions, but there are things that we know we can improve on. There's lots of suitable habitat in BC, but it's very patchy, and it's likely to, but we'll see more of it as climate change progresses and things get warmer. Finding those habitats, though, is something we need to work on, and we can do that through species distribution modeling, through rapid site selection, and potentially through eDNA. So those are all the different sort of aspects of spread that we're working on. And now I want to do some impact stuff for you guys. And this is really the so what, why put all this effort into finding green crab in the first place? I'll start just by giving you a quick overview. Impacts of invasive species can happen at genetic level, the individual level, all the way up to the ecosystem level. I'm going to talk really quickly about population and community level impacts of green crab, and then I'm going to focus on one particular example of ecosystem impacts in British Columbia. So for population and community impacts, we're relying a lot still on studies that were done in California when green crab arrived there. Long-term tracking of species in the intertidal, Bodega Bay, I think specifically in this case, showed that when green crab arrived in 93, 94, all of a sudden there was this huge downturn in the population densities of Hemigrapsis, which is a native shore crab species, and Nutricola, which is a clam species, an infaunal clam. And I want to draw your attention to that pattern with Nutricola, because it's one of the things that green crab are kind of infamous for. They are, again, just really, they're generalist predators, but they're really effective shellfish predators in particular, partly because they have these two different claw sizes. And so they're quite happy eating clams, and we have quite a few clams. We have a lot of clams. We have a lot of industry that relies on clams. And this is something that we're going to be doing more on, and we need to ask more questions about. But certainly the impact of green crab on our infaunal clam populations is a big question. And research from both south of the border and elsewhere in the invasive range of green crab all indicate that green crab are not great for native clam populations. We also have a lot of questions around competition with native crab species. Like I said, or showed in the previous slide, hairy shore crab, or Hemigrapsis species, definitely seems to take a hit when green crab populations come up. Inverse of that, we see red rock crab tend to outcompete and or predate on green crab. So this is actually probably what excludes green crab from rocky habitats. Red rock tend to hang out in rockier habitats. That's where they're sort of more specialized as predators. And so because they tend to outcompete and eat green crab don't occur in rocky habitats as frequently. The one question sort of in between those two, the thing that we're sort of the most unsure of and would like to know more about is what the interaction is between Dungeness crab and green crab is. And this is for lots of reasons, not least of which Dungeness is obviously an important fishery. There's some studies that show that green crab outcompete small Dungeness crabs, but whether or not that would scale up to population effects is something that we haven't detected yet. And so those are sort of population or species interactions that we know a little bit about. And now I want to focus on the effect of green crab on an ecosystem, specifically eelgrass habitat. Eelgrass is a really important species in British Columbia and around the world. It's a nursery habitat for things like salmon and juvenile Dungeness. Herring lay their spawn on eelgrass. It just supports a huge biodiversity and is sort of this critical habitat that links freshwater, terrestrial, and marine habitats. On the east coast of North America, a number of studies had come out showing that increasing populations of green crab were really bad for eelgrass. So either anecdotally, they saw these huge declines in eelgrass as green crab populations came up, or in a few cases, they actually ran experiments, which is the picture on the right there, putting in enclosures, and then the green crab would just sort of tear up all the eelgrass while they searched for their bivalve prey. So this is obviously a concern on the Atlantic, and I wanted to know if the same pattern would occur here in the Pacific. So this is something I did while I was at SFU doing my PhD, and I asked the question, or we asked the question, do green crab have a negative impact on eelgrass habitats in British Columbia? This is the paper that came out of that. It is behind a paywall. If you're interested and want to dig into this more, please just get in touch. I'm happy to share it because, yeah, I think it's important work, and usually people have questions. So please let me know if you'd like a copy of that paper. So to do this experiment, we set ourselves up in a little eelgrass bed in Barkley Sound where there were already a low density of green crab. We had 4 treatments, no enclosure, so the eelgrass was exposed to just ambient green crab density. We had an empty enclosure, which had no green crab, and low density and high density enclosure. These are what our enclosures look like. So they were fairly big, and we left them in for a month and let green crab just do what they were going to do. And on the bottom there, I've got a picture of my colleague Fiona Francis and co-author doing all the hard work of actually counting the eelgrass by snorkel while I just hung out on a boat. So I always want to take a minute to just props to Fiona for the amount of physical labor she put in on this project. And here's the big picture, what we found. Overall, there's this declining in eelgrass across all treatments. And that's because we were conducting the experiment in August. And so there's just a natural sort of senescence happening with the plants anyways. But what that really emphasizes is what's going on with that high density treatment. The eelgrass decline is way above normal levels there. And even within 2 weeks, we saw a pretty extreme response. So it doesn't take a lot for eelgrass to be negatively affected by high densities of green crab. Inevitably, that leads to the question of how high is high. And is 5.6 crabs per square meter a lot? I would direct folks to the few sites, those hotspots that I mentioned earlier, where we're pulling up more than 1,000 crabs a day. That in a site like that, 5.6 crabs per meter squared isn't really hard to imagine at all. Fortunately, there aren't a ton of sites like that, but it does mean that we need to be able to find those sites and protect those sites before they turn into green crab hotspots, especially if they have things that we care about, like eelgrass that we want to protect in those habitats. This figure, just to quickly cover it, is the catch per unit effort, average catch per unit effort of green crab over time. It stops in 2019, so Haida Gwaii is not yet on that list. And you can see, although Salish Sea and North Central Coast are, very low or zero, what's going on the west coast of Vancouver Island is not something we want to see repeated elsewhere on the coast. So in conclusion, ecosystem impacts, I believe, are definitely possible at high densities. Eelgrass supports a huge diversity of organisms on our coast. It goes right from the little tiny inverts that feed and support, our juvenile salmon and other species. And from there, you can scale all the way up, right? That's the sort of trouble with ecosystem impacts, is we don't know how far they scale up. And so that's something that we're interested in, for sure. I'm going to wrap up for questions in a minute here, but before I go, I just wanted to quickly cover off how you guys can help. I'm sure you're curious. First and foremost, please report sightings, especially if you see green crab in the Salish Sea, on the North Coast, Central Coast, Haida Gwaii. those areas where we're seeing them expand into. So this is the email address that you can send your sightings to. Please take photos as well as GPS coordinates. We need to be able to confirm the ID, of course. And just to reiterate, the main things that you're looking for are those 5 spines on the side of the carapace, the 3 spines between the eyes, and don't use color. You can also, of course, support local organizations working on green crab mitigation, and there are lots of them, including, of course, you yourselves at the Invasive Species Council at BC. And I'm sure most of you are familiar, but I might as well say it: clean, drain, and dry. It's not just a freshwater thing. It really matters for marine environments as well. The bottom of boats, they just can carry a lot of biodiversity. And so the more you can clean, drain, and dry, the healthier our marine ecosystems will be for sure. So without further ado, just some quick thanks. I've highlighted Tom Theriault, Isabel Cote, and Fiona Francis up at the top there because they were my co-authors on that eelgrass paper, and I just wanted to thank them again for their hard work on that project. I also have pulled out Rennie Talbot's contact information. Rennie is the person sort of leading the management side of things for green crab at DFO. So if you have a question that you think is more in that wheelhouse, you're more than welcome to get in touch with him directly. Otherwise, I'm happy to forward any questions on as well. And you're more than welcome to get in touch. Loads of great colleagues at DFO who have done tons of work and let me present on the sort of stuff they do, as well as lots of external collaborators that I'm super grateful to have the opportunity to work with on this particular issue. So yeah, with that, I would love to take any questions that you might have, if there's time.
Yeah, there's definitely time, Brett, and just a big thank you for your presentation. It was fantastic. I definitely took some notes myself, and I'm sure lots of our attendees did as well. And also thanks for your energy and enthusiasm on this topic. We have quite a few questions, so I will start with the first question and work my way through as time allows. And as I mentioned before, if there's not enough time for all of the questions to be addressed, there will be an opportunity to pass your question along to Brett. So the first question is from Robin Taylor. Given that ballast water seems to be the vector for a large number of known invasives and potential carrier of lots more, what is being done at the federal level to manage this water? Detection once it is introduced is only part of the solution. Absolutely,
that is absolutely true. The question is probably best answered by somebody with with a better connection to Transport Canada, because this is definitely something where you're talking about a collaboration between DFO and Transport Canada. We do have ballast water exchange areas that are well offshore. And so the idea is that you dump your ballast way offshore in an area that is definitely inhospitable to anything that might be in that ballast water before you come inshore. And I believe in Canada, those are pretty well established. It's a bigger problem in places where you don't have as much enforcement of rules like that. But I'm not going to go any further than that because I would be talking outside of my immediate area. But it's certainly a priority. Another priority is the sort of stuff that grows on the bottom of those boats and how we can minimize that as well. But those are relationships between DFO and Transport Canada that are ongoing.
Great, thanks, Brett, for that. The next question is from Matt. Matt is wondering how trapped European green crabs are disposed of.
Okay, great question. So obviously we want to do it as humanely as possible. If we are dealing with a huge volume of green crab at once, they're killed basically by smushing as expediently as we can. If we have the ability, our preference is to freeze them, but that's really only practical if you're dealing with very small numbers of crabs. This is also a preference because it allows us to do genetics work if we take them back to the lab and freeze them. So it really depends on whether the volume of crab that we're dealing with.
Yeah, I would assume if you had a big bunch, you'd need quite a lot of freezer space for that, which isn't always available.
Yeah, exactly.
Next question is from Nikki Wright. Could this PowerPoint presentation be made available for outreach purposes to non-government organizations?
Probably for outreach or for outreach material, my suggestion would be to get in touch with Rennie first, only because they are actively developing outreach material. So it might be a bit more tailored for whatever purposes you're thinking of. But that said, if there's something that I can provide you with that something specific from this talk that you would like, I'm more than happy to share. Just get in touch. And of course, the presentation itself is available. Perfect. Or recorded, I should say.
And next question is from Stefan. Hopefully I'm pronouncing that right. To what extent do you engage citizen scientists in detection?
Great question. So that is a growing sort of portfolio, shall I say, for Rennie and Patty. They are engaging a lot of different community groups, doing a lot of training, which is best done in person, and that has made 2020 quite a challenge for Patty, but they've done a great job. They've engaged lots of different community groups, trained them on how to ID green crab, and in some cases that's proceeded on to actively trapping or surveying for them as well. Again, I would hate to misrepresent the work they're doing. It's very involved and detailed, and you're more than welcome to get more details off of Rennie and Patty on what they're doing. But yeah, it's a growing program, both in the Salish Sea in particular, but increasingly up on the North Coast and Haida Gwaii as well.
Yeah, I really enjoy being a part of citizen science myself, but being in the interior, I can't really help on the European green crab front, but I definitely get out and love,
it's fantastic. And I will add that a lot of what is being done on the citizen science front is we're taking a bit of a lead from the crab team in Washington. So they're the Washington Sea Grant crab team. If you happen to be listening from the other side of the border, they are fantastic, and they really advanced the way we do citizen science for green crab. They led the charge, and so I just want to give them credit as well, because they do a lot of citizen science.
Yeah, that's great to hear. Now the next question, I know in your presentation, Brett, you did talk briefly about eDNA, but Bronwyn has a question. Are there any agencies in BC actively using eDNA for early detection?
That's a great question, and I don't know the answer to it, unfortunately. eDNA has sort of its origins from an invasive species perspective, has its origins in freshwater. And so I'm sure that there is eDNA being applied by somebody somewhere to things like the Dracenid mussel issue. I'd be surprised if that weren't the case. It's certainly the case elsewhere in Canada. eDNA is a very popular tool for that problem. Whether there are actual outside of government agencies using eDNA for aquatic invasive species, I'm not sure. Certainly there's a long way to go to really get it tailored for marine environments, just because it kind of came out of freshwater. The methodology is originally from freshwater systems, but yeah, it's being used more and more, especially for invasive species detection, just because you can get such high throughput.
And then Janet is wondering if you use data sets like shoreline when looking at hotspots.
Yeah, absolutely. Shoreline has been shore zone. Yeah, shore zone is I can't state how much time I spend looking at shore zone stuff. It's incredibly important to the species distribution modeling work that we're doing. I'm certainly using it directly in that rapid response species distribution model that Tom and I are developing. I use shore zone all the time. It is extremely helpful. It's sort of it's one of the best, if not the best, and most comprehensive sources of data that we have on an area that is generally very understudied. The intertidal is tricky to get to, tricky to quantify, and so having all of that data is fantastic. Yeah, definitely use it.
Great. And then we have another eDNA question. So Nick is wondering if European green crabs shed lots of eDNA, and how viable of a method are you finding it?
In terms of how much DNA they shed into the environment, I'm not sure how that would stack up against any other species. One thing I will say is that all crabs molt, right, as they grow. So you do get this periodic shedding of a whole bunch of material. I imagine that would certainly contribute to the overall quantity of eDNA in an area, especially in the summer when they're molting at a really high frequency. Also, I mean, the more you eat, the more you poop. So there's probably lots of that in the water as well, especially again in the summer when they are, eating lots, breeding lots. So you got eggs, you got larvae. There's lots of different sources, and I suspect in the summer in particular, you would find the sources of eDNA would be really high. As to how well it works, in general, I had always thought of eDNA being a presence-absence kind of thing. So I'm personally really blown away by and excited by the potential for it to actually link up to a bit more of the abundance side of things. That said, I'm not a geneticist, so maybe I'm just easily impressed, but I thought that was cool, and I think has tons of potential. But yeah, not a geneticist, so.
Awesome, great. So many good questions today.
Seriously,
we've got another question from Becky. Becky is wondering what measures are taken during fieldwork to ensure other invasive species at sampling sites are not being spread on sampling equipment from one site to another. There is an interface between some of the confirmed green crab sites with confirmed invasive intertidal Spartina grasses.
Good question about Spartina or Spartina. I'm going to think about that some more. So we definitely know that green crab really like that sort of salt marshy sort of habitat. It works well for them, especially when they're small and establishing. So the interface between those two as invasives is, there's a lot there that's potentially really interesting. But getting to the actual question, which is what are we doing to make sure we're not contributing to the spread? Obviously, that would be really embarrassing and very, really not good. So we do take precautions to make sure that we are not spreading invasive species around. In between all of our sampling trips, all of our gear is washed in fresh water and stored and dried. So we aren't moving species around on our gear that way. And then we also take a lot of care with our vessels. Every time we put them in and out of the water, we're checking and making sure that green crab are really good escape artists. So you really do have to check all the little nooks and crannies of your boat and make sure you're not taking any green crab with you. Beyond that, I suspect, I haven't done a lot of the intertidal work, but I suspect in between intertidal sampling trips, doing the same thing, making sure we're not transporting mud between sites and that sort of stuff. So certainly it's something we're all very aware of, and we take care not to move species from site to site, because that would definitely not be a good thing.
Yeah, definitely. I know if I'm out with volunteers doing a weed pull, we definitely have to, clean our gear and use boot brushes and make sure that you're not taking any invasive hitchhikers with you when you leave the site. It's totally important. Yeah,
one of the upsides to marine invasive species is that they are, in general, very susceptible to freshwater. So it does help that we have a pretty good way of making sure that nothing survives between trips, as long as we give everything a really good freshwater rinse.
Perfect. And our next question is from Adrian. And Adrian is the marine ecologist with the Maca tribe in Nia Bay, Washington. At any locations, have you tried using recreational shrimp traps?
That's a good question. So there's a lot of interest in coming up with other ways to trap. So in science, we rely on those Fukui fish traps for a whole bunch of reasons. First of all, it's standardized both here in the Pacific, and it's the same type of trap they use for green crab monitoring on the East Coast as well. So it's standardized for us within science, which makes it really easy to do things like compare catch per unit effort. So from a perspective, we're sort of sticking, that's the horse we picked and we got to run with it. That said, it has drawbacks, and there are studies that show that it is not effective trap for catching green crab en masse. And other types of traps, including, I believe, prawn traps, are likely better, or possibly better. That work is ongoing, and there are folks that are sort of testing out different trapping mechanisms and trapping methods to find the one that's best for detection and eradication. So again, you have sort of different goals for management versus science.
Great. Okay, we've got time for a couple more questions. Tim is wondering, is there any possibility? I'm just getting a bit of feedback, Brett. I don't know if it's on your end. Perfect, thanks. So Tim is wondering, is there any possibility of controlling the species? For example, could a major trapping effort contain and slow or stop the spread?
Okay, you can hear me? Yeah.
Yeah, it's great.
That's an excellent question. I'm really glad you asked it because, yeah, that's something that I should definitely make clear. So for green crab, we don't anticipate that there's really a possibility of, say, eradicating them from the coast. They are so widespread at this point, and of course that's a US and Canada thing. So eradicating them entirely is sort of beyond scope, but that doesn't mean that we can't control them location by location. That's why we are targeting this response effort into the Salish Sea and Haida Gwaii right now, where the populations are still small. For those invasive species nerds, the term early detection and eradication might be familiar to you, and that's exactly what we're trying to apply, right? Try to detect them early and get rid of them while the population's still small in localized areas, and we think that is completely doable. So that's definitely the goal for us. Yeah.
Yeah, and that's why it's so important that people report sightings of invasive species or something that seems out of the normal to them. And then that way there can be that early detection, rapid response happen with new invasives. Next question is from Janice. What are the natural predators for European green crab, and are they edible by humans?
Yes, okay. Two good questions. We get this a lot, and yeah, people are very curious about predation for green crab. So it's all anecdotal at this point. Like, people have seen this species eating green crab. To what extent our native predators are taking out green crab, we can't say. But we do have, we have visual confirmation that sea otters and river otters will eat green crab. Gulls will eat green crab. I suspect, but don't know, that bears would also eat green crab if they were small and underneath a rock, like they do with other shore crabs. I don't think bears are too discriminate when they're just flipping over rocks in the intertidal. So there's a number of species that we suspect would opportunistically eat green crab. Certainly our native crabs will eat green crabs, and helpfully green crabs eat green crabs. They're actually a pretty cannibalistic species. The larger ones will go after the smaller ones. And so those are all the things that are eating green crab. In their native habitat, they're also controlled by parasites, but those parasites aren't found here as well. Thankfully, we wouldn't want an invasive parasite on top of an invasive crab. But that's what controls them. That's one of the main sort of population control mechanisms in their native environment is parasitism. For eating them, right now that hasn't been looked at by Health Canada. So while technically edible, they're not approved for human consumption, and that's sort of where we go with that is, and besides, why would you when you have all these beautiful Dungeness crabs and red rock crabs on this coast to eat?
And we have time for one more question, which is from Nick. Has the public been reporting European green crab?
Yes, they've been a huge ally in finding green crab. And I mean, one great example is, of course, Haida Gwaii. It was detected by somebody who I think works for Parks Canada, but was just out on a walk as a member of public and found green crab. So absolutely, the public has been super helpful in detecting green crab. it's a very complex coastline, and humans are distributed quite sporadically around, and we can't get everywhere. So having people detect green crab and find them and let us know that they're there is really, really helpful, because then we know exactly where to go put our traps, which is one of the biggest problems. So yeah, absolutely. Please report green crab if you see them, especially if you see them in the Salish Sea, Central Coast, North Coast, Haida Gwaii.
Okay, well, a big thank you to all the attendees for your great questions. And on behalf of the Invasive Species Council of BC and all those that are listening, thank you so much again, Brett, for sharing your expertise with us today. That was an amazing presentation. We will be emailing all attendees a link to a short survey. Please take a moment to complete it and provide your feedback and ideas for future webinars. If you enjoyed this webinar, please consider supporting ISCBC's work by joining us as a member. Members are offered discounts on paid events and training courses, among other benefits. Visit bcinvasives.ca/join to learn more. Also, if you live in BC and you'd like to make an impact in your community and are between the ages of 15 to 30, consider volunteering with us. You can find out more at bcinvasives.ca/volunteer. And thanks everyone, and we'll see you next time.